All right, so now we're going to focus in on chapter 5, where we're going to be looking at microbial metabolism. So we need to define metabolism. Metabolism is the sum of all the chemical reactions that happen for a cell. And so there are two different types of metabolism. There's catabolism. This one is a degradative and exergonic type of metabolism, meaning that it's going to break things apart and it's also going to release energy. Some of these are also what we call hydrolytic, meaning that they have water as an input. Water goes in to break the molecule apart. So this is also what we would consider a hydrolysis reaction. So if you look here, you have the polymer. Polymer means it has many parts to it, and we're going to add water. When we add water to this reaction, it's going to cause the molecules to break apart. When they break apart, they're going to release energy. This breaks the polymer down into their building blocks, and in this case, we're looking at, and this gives you the example with the monomers. Now, again, you need to recall that water is an input here on some of these types of reactions. Water is going to go in, therefore it's a reactant. On the other hand, we have anabolism. Anabolism is going to be a biosynthetic and endergonic type of reaction, meaning that we are going to build something and also we're going to put energy into the reaction. Now, some of these are considered dehydration reactions because we are going to be taking water out. So water is an output, meaning that it is a product of this type of reaction. So we're going to take a monomer plus a monomer. Okay, when we do this, we're going to add energy in. So we're going to put energy in. This energy is going to be added into the bonds, creating the polymer, and water is going to come out of the reaction. All right, so we have kind of like a little quiz here to kind of look at this. We have a monosaccharide with a monosaccharide. We are then going to make a disaccharide. Now, meaning the disaccharide is going to be larger. And if you'll notice out here that water is an output, it comes out of the reaction. So the first question here is this dehydration or hydrolysis? Since water comes out, this is dehydration. Also, is this anabolic or catabolic? Since we're building something, this is going to be biosynthesis and it's an anabolic reaction. Also, did energy go in or did energy come out? Well, if we're going to build something, energy has to go in. So is it an exergonic or endergonic reaction? In this case, it would be an endergonic reaction. So this one reaction, if you notice, we could characterize it in multiple ways, depending on which part we're looking at. Dehydration, hydrolysis, anabolic, catabolic, exergonic, or endergonic. All right, so what we're going to be looking at here is this whole idea of energy is going to be in the form of ATP, adenosine triphosphate. One thing to note though is this is a coupling reaction. When we see here, it means that we can build it, but we're also then going to break it down. So energy is going to go in and energy is going to come out. This is going to be a series of catabolic reactions followed by anabolic reactions. So if you follow the flow chart here, we can see that we have a complex molecule coming in down here at the bottom, such as a starch, protein, or a lipid. All right, so this would be something that you might have taken in. Like for us as humans, we would eat it. Organisms sometimes will consume them as well, and they want to break them down into the building blocks. This is the catabolic reaction. They're going to break down the pieces. When they break down the pieces, heat's going to be released, and now we have some smaller molecules, and energy gets released in the form of ATP. This ATP now can be used with these building blocks to make their own complex chemicals, complex macromolecules. So if that particular molecule or if that particular cell needs a carbohydrate or it needs a protein, it can now use this energy and these building blocks to then build it. This is the anabolic side. In this case, we're putting energy in, therefore we're going to get ADP and a phosphate out that can be reused in the form of making ATP again when we do a catabolic reaction. So this is a type of cycle that happens in the cell. Now, one thing to note with this is that on both sides of here, heat gets released. Energy transformations are not 100% efficient, meaning that some of the energy is going to end up being kind of lost in a sense. It's not actually lost forever, but it's been transformed into heat. This heat is normally not usable, all right? We can't necessarily use it as that cell. And so this is going to be some unusable conversion. So the concept here when we looked at this chart is that catabolic reactions, since they release energy, are going to drive anabolic reactions in order to give energy to build those new substances. So if you look here, the catabolic reaction takes A and B, and we're going to break it down into A plus B. Energy gets given off. It's an output. We then use that same energy as an input where we take C plus D, and we're going to combine it into CD. All right, so the catabolic reaction is going to drive the anabolic reaction. So when we're looking at metabolic pathways and chemical reactions, these a lot of times require enzymes. These enzymes need to be present, but they also need to be functional. Now, one thing to note about enzymes is that they are normally proteins. It takes many different enzymes in order to catalyze all the reactions that are within a single cell, and this includes even our microbiology cells, our bacteria. However, cells do not make large quantities of these enzymes. These enzymes can be reused, so we don't need to make a lot of them, but we do need them present and functional in order to complete these reactions quickly and efficiently. So what is an enzyme? An enzyme is an organic protein catalyst. All right, so it's made out of a protein, and the whole idea here is being a catalyst, it means that it speeds up the reaction. It makes the reaction go faster. Now, this is going to be because it lowers the activation energy. It means that the enzyme makes it where there's less energy that has to be put in order to drive the reaction. So if you take a look here at this picture, you'll see that the normal reaction takes place in the red. Okay, we have to get a certain amount of energy before we actually see that the reaction will take place. All right. However, when the enzyme is present, you'll notice it in the blue. In the blue, we see that it still has to require some energy, but it does not have to go near as high, and then the reaction still happens. And so because less energy is required from the get-go, it can actually make the reaction happen quicker. So the major concept here is that enzymes lower the energy of activation. Now, enzymes, because they are a protein, they have a three-dimensional shape. This three-dimensional shape is what makes them functional, and this shape also has an active site. The active site is where we're going to see the reactant, or what we call the substrate, go in to the enzyme. Now, these are very specific, all right? So they have a very high specificity. When we look at this, think about your house. Your key hopefully only works on your front door, all right? They would freak you out if you went to your neighbor's house and you put the key in and it turned and you could get access into their house. Okay, so when we look here, we want our keys to be very specific to our home. Okay, the same thing here. These enzymes are going to be very specific to one particular thing. We also see that a lot of times enzymes are going to end in ACE. Okay, so it'll have the substrate or what it's acting on and then ACE on the end. So some examples here. So the first substrate here is urea. The enzyme that works on urea is called urase. Okay, because it ends in ACE. Another substrate is lactose. Lactose is a type of sugar, and the enzyme that acts on lactose is lactase. Okay, so an individual who's considered lactose intolerant, it could be that they're lacking this enzyme to break down that sugar. All right, and so when we look here, the substrate is going to fit into that active site, kind of like in a lock and key type mechanism. So if we look at this, enzymes a lot of times are classified based on the type of reaction that they catalyze. So if you look at these ones that talk about oxidoreductase, these guys are going to do an oxidation reduction reaction in which oxygen and hydrogen are either gained or lost. One big example here that's in red here is the cytochrome oxidase. This one is going to be really important in cellular respiration. Another one is transferase. Transferase is going to transfer functional groups. It could be an amino acid, an acetyl group, or a phosphate group. One of the main one that's important with the phosphate group is if we add phosphates or move them, that's going to help us with ATP, because ATP is adenosine triphosphate. So kinases are an example of this when we see them in cellular respiration. There's also the hydrolase. This one is going to do hydrolysis. It's going to add water. Lipase, sucrase, any of these that are going to help add water to break things down that we consume or that an organism would take in. Lyase is going to be the removal of groups of atoms without the use of water. Okay, so they would use this in a little different way. We also see isomerase. This is going to rearrange the atoms within a molecule. This is going to make it where it looks different. The same structures are there, but it looks different. And then ligase is one that you may have learned about in general biology that is important in helping hook things together, join things together. Ligase is important on that strand in the DNA replication where it's adding the Okazaki fragments and they need to be added together, and ligase is the enzyme that helps do that. So there are some things that might need to be present in order for an enzyme to be functional. Not only does it have to have that 3D shape, but it also may have to require the presence of one of the following. One is called a cofactor. Cofactors are inorganic metal ions. These are going to be things like iron, copper, zinc, those sort of things that are considered inorganic. Or it could also need the help of coenzymes. Coenzymes are going to be organic molecules. Some examples of these are NAD+, FAD2+, and CoA. We do see these as being really big factors in cellular respiration that are needed in order to help the reaction to continue. A lot of times these are synthesized from vitamins, and this is one reason why vitamins are so important in the diet. It shows you that you have the enzyme. However, the enzyme in this case is inactive. When you add the coenzyme here, that is going to make the lock and key type structure where the substrate will then fit in. So it has to have that coenzyme or cofactor present in order for it to be able to actively bind to its substrate. So there are some selected vitamins and their coenzyme effects in this particular table. So if you look here, some of them are highlighted for you, like riboflavin. Flavin is going to be an active in electron transfer, so like the FAD. That's why it's called flavin adenine dinucleotide, what NAD is going to be part of. And then you can also see the coenzyme A. So how does the enzyme work? When we look at the enzyme, we're going to take the enzyme and we're going to add it to the substrate. This is going to be the reactant. Okay, when the substrate is added, this creates the enzyme substrate complex. Once the enzyme substrate complex has been completed, we see that the enzyme may change shape or do something where it's going to then create the product. All right, so it's going to change the substrate into the product. But if you'll notice over here, it still says enzyme plus product. The enzyme is not changed. The enzyme can now go and get a hold of another substrate and continue to do its job. This is why we don't have to have very many of those enzymes present, because they can continually work. They're not changed by the reaction. All right, so going back to our terms here, we have a little section here that says, is this anabolic or catabolic? All right, so the substrate went in, okay, as one piece. It comes out as a product in two pieces. All right, and since we broke it apart, this would be an example of a catabolic reaction. There are some factors that influence an enzyme's activity. There are some things that will actually slow down or speed up the rate of how the enzyme works. I mean, think about yourself. Every one of us has like a certain time of day that we work best in. Some people are morning people. They hop up out of bed and they can get things done real quick, but they start to lose steam maybe in the afternoon. Other people are night owls. They want to stay up super late, and that's when they're most productive. Some people like myself, we're more midday people. Okay, it's hard to kind of get going in the morning, but once we get going, it's pretty good, and then we start to slow down when we start to get tired. But when we look here, enzymes kind of have these same types of things. They have certain conditions that they work best in. All right, and so one example of this is temperature. Okay, they're going to have a very optimal temperature. When we are too cold or too hot for the enzyme, it's not going to function properly. Another one is the pH. Okay, the pH can also be where it's too acidic or too basic. It's going to have an optimal pH that it prefers. Okay, in this case, a lot of times most of the enzymes like to be more around neutral. Okay, but when we look here at these particular diagrams, the enzyme's activity can go up and down based on the movement of the temperature or the pH. Now these two have little stars next to them because these two can actually cause the protein to unfold and denature. This makes the enzyme no longer functional. Remember, the proteins have their particular structure where they have the string of pearls. They then have the beta sheets and alpha helixes. Those then wad up on themselves to cause the globular protein. And when that unwinds back to its secondary or even primary structure, that enzyme is no longer functional. Some other things that can influence the enzyme's activity is the substrate concentration. The more substrate that is present, the more it's going to react with, making the reaction be faster. Also, the enzyme concentration, the more enzymes that you have, the faster they may work. However, if there's too many of them, then of course it could cause some problems too, slowing things down. So when we look at substrate concentration and enzyme concentration, we're going to see that there's going to be an increase, but then there'll be a plateau. That plateau will happen where it's at the max that it can work and function. There can also be what we call presence of inhibitors. And we'll talk about inhibitors on the next slide. So if you look here in this particular example, when heat increases, collisions between the enzyme and the substrate start to increase. Okay, and so this is going to be where they increase up to a certain point until it gets too warm in this case, and then the enzyme starts to denature. It starts to unfold. Same thing here with the pHs. If we are on either side of that pH, if it's too acidic or too basic, the enzyme will denature as well. So when we are looking at inhibitors, this is really important to know if they're actually taking up the active site, the place the substrate should fit into, or if it's going to fit into what we call an allosteric site. So some inhibitors are going to prevent the product from being formed. If they are a competitive inhibitor, it's going to compete with the substrate for the active site. So if we see the picture over here, this is how the enzyme should look. This is what we should see that the substrate fitting in. It's been outcompeted by the inhibitor. The inhibitor is taking up the space, which makes it where the substrate can no longer attach. On the other hand, so I like to give the example like in a parking spot, where you see an empty parking spot and you whip around, you're going to try to go down to get it, and when you get there, now somebody else has taken it. They have taken the place, and therefore you can no longer get in. Okay, and even worse, if you didn't notice, it was a small car or a motorcycle that was there to begin with, and this site was never even open for you to begin with. On the other hand, there's the non-competitive inhibitor. The non-competitive inhibitor sets in an allosteric site. It's in the vicinity of the active site, but it causes the protein to change shape. When it changes shape, the enzyme can no longer bind to the substrate. So in this example over here, it shows you that this is a non-competitive inhibitor. It is going to bind to the allosteric site. This causes a change in the shape. So if we go back to the parking lot example, this would be like if, especially here in New Mexico, I don't know in some of the places where you guys are at, but the big pickup trucks that people drive sometimes, if they don't park right and they're over the line, they may change the parking space just enough where your car can't fit. Okay, it changed it just enough in the shape in order for you to not be able to fit. Another example would be if there's a cart present. Okay, if the cart's there, now that could easily be changed, or you kick somebody out and say, hey, go move that cart. But if you're by yourself, that may make it where the parking space can no longer be used. All right, it changes the shape just enough, but it's not actually taking the space of the active site. Now, cells can actually use this idea of inhibition in order to do what we call feedback inhibition. This is a normal mechanism that cells can use to help control the amount of product that they actually make, because we only need so much of the product to be around. If we don't have any more of the substrates left, or if we have too much of the product, we don't want those enzymes to continue to work. We don't want to waste that energy. And so because of that, we want to shut them down. So an example of a feedback inhibition is where you start with substrate A. Substrate A is going to be taken in by enzyme A, okay, and we're going to say this is ACE, because remember enzymes in an ACE, and it creates the product B. Now B is going to then be turned into C. In order to do this, it's another enzyme that's present that we're going to call BACE. C is then going to turn into D, which is called with an enzyme CACE, and then D is going to turn into E. Now E is our ultimate product that we want to get to, and DACE is the enzyme that gets us there. This is a series of steps. So once we have enough E, okay, in the cell, we have enough E present, we want to shut down this pathway. In order to shut this down, this final product of E can temporarily inhibit the first enzyme. In this case, it's going to go and connect to an allosteric site on AACE. This is going to stop the sequence, okay, because it changes the first enzyme, therefore we can't make B, then we can't make C, then we can't make D, and we don't get to E. However, when we don't have enough E, this E will break off and the pathway will turn on again. So if you look here in this picture, it shows you that E connected to the AACE. It's now inactivated, and now A can no longer attach. So note that each step requires its own specific enzyme, so this is a type of negative inhibition. However, it is a reverse condition. We can turn it off. So too much product, the pathway gets shut off, and not enough product, the pathway gets turned back on. All right, so we can reverse this and go back and forth. So this is going to show you again how this works, where the cells can regulate their metabolic pathways. Here's an example again of a feedback inhibition. Remember though, it is reversible. That is important. When the final product accumulates, it's going to be the one that binds to the first enzyme. This is going to shut off the pathway because now the first enzyme has changed shape. Therefore, it can no longer attach to the initial substrate. Now, this means the pathway is now turned off. When we need to make more of the product though, we'll take the product off and we can turn the enzyme back on, causing the product to then be made through this process. All right, so now let's look at how cells produce this energy, and we're going to generate here ATP. How we're going to convert ADP into ATP. Now, ADP is adenosine diphosphate, and ATP is adenosine triphosphate. When we're fusing the phosphate onto ADP, energy is required. This energy is then stored in that last bond. It's called a high energy bond on that last phosphate, and now we have ATP, adenosine diphosphate. Now, one thing we're going to look at is we're going to compare substrate level versus oxidative phosphorylation. Okay, phosphorylation is just a big fancy word that just means adding a phosphate. All right, so when we're adding a phosphate to something, we would say this is phosphorylation. Now, there are different ways we can add this phosphate. Okay, there's the substrate level and there's the oxidative. So with substrate level phosphorylation, this is where a substrate donates the phosphate group. It'll donate it to ADP in order to form the ATP. This does not produce as much ATP as oxidative phosphorylation. If you'll notice, this does also require an enzyme. So the enzyme comes in, it connects to the substrate, it transfers the phosphate from the substrate to ADP, and what comes out of the enzyme is the product plus ATP. And so since the enzyme can only create one ATP at a time, this process is not going to be very efficient. Now, glycolysis is an example here of a type of substrate level phosphorylation. Okay, when we take glycolysis, we're going to take a glucose molecule in. Glucose is then going to be broken down. It's going to be halved. Okay, glucose has 6 carbons to it. It's going to be halved into a 3-carbon molecule and another 3-carbon molecule. These are called pyruvates or pyruvic acid. In this process of breaking apart and actually physically breaking the bonds, energy does get released here. This energy that is released is going to create 2 net ATP with substrate level phosphorylation. Now, you'll notice though, guys, ATP has to go in. This is where it kind of comes. You know the saying, it takes money to make money? The same thing happens here. It takes energy a lot of times to make energy. So we see that 2 ATP comes in and 4 ATP comes out. But since we used 2 to begin with, we only gained 2 ATP in this process. And this is the first step of cellular respiration, which is known as glycolysis. Another place where substrate level phosphorylation takes place in this process of cellular respiration is in the Krebs cycle, also known as the citric acid cycle. Now, I'm not going to have you memorize every little step here, but I do want you to see that enzymes are important in these different levels. And in this process, you're going to see that 2 more ATP are going to come out of this. Now, remember from glycolysis, we had 2 pyruvates or 2 pyruvic acids that came out. Each of these are going to go into the Krebs cycle. So when it turns for first time, it's going to create 1 ATP. When it turns with the next pyruvate, is going to create 2. So we're going to get 2 ATP again from each glucose molecule that we started with. So now we're up to potentially 4 ATP. Now, one thing to note in both of these, you will notice that there are some special carrier molecules who are present. You'll notice that in some of these reactions, we're going to take NAD plus and it's going to be turned into NADH. This happens in glycolysis and it happens in the Krebs cycle. Also in the Krebs cycle, you're going to take FAD and it's going to become FADH2. They're picking up hydrogens, which also means they are picking up electrons. This is going to be important when we get to the oxidative type of phosphorylation. So in oxidative phosphorylation, we see some redox reactions are going to help produce ATP. Now, in a redox reaction, this means we're going to transfer electrons. And this also means that we're going to be transferring hydrogens in the process as well. This transferring that takes place in order to produce ATP is going to be done via chemiosmosis. So electrons are energy carriers. They get removed from organic molecules and they are going to be transferred to what we call the ETS. This is the electron transport system. Sometimes it's also known as the ETC, known as the electron transport chain. These electrons with these hydrogens are going to be transferred by NAD and FAD. These are going to be like the taxi cabs for them. They're going to send them from where they were made, whether it's in glycolysis or the Krebs cycle, and it's going to take them to the electron transport system. Now, oxidation is the loss of electrons in a hydrogen atom. So an organic molecule will become oxidized when it loses electrons or hydrogens. It then becomes reduced when it gains the electrons and hydrogens. Now think about this. Remember, electrons have a negative charge. So when they gain a negative, that's why they become reduced. Okay, this is going to be where NAD and FAD get reduced to NADH and FADH2 because they accepted the electrons. All right, so if you look here in this example, it shows you the reduction part where we're going to transfer over, where we see that they've gained the electrons, and then the oxidation part is when they've lost the electrons. Now these electrons are going to be dropped off at the electron transport system. So when we're looking at oxidative phosphorylation, this is going to occur in the plasma membrane of prokaryotes. Okay, so when we see that the prokaryotes, remember, they don't have organelles, so they don't have mitochondria present. And so because of that, this process still can take place, but it's going to take place in their plasma membrane. In eukaryotic cells, like in protista, fungi, and animals, we see that this process takes place in the organelle, the mitochondria. Now, oxidative phosphorylation is going to produce a lot more ATP than substrate level phosphorylation. And if you'll notice here, it's because it goes through a series of steps. And each of these steps, we see the electrons are transferred, and when they are being transferred through this oxidation and reduction reaction, lots of energy ends up being released. There's 32 to 34 ATP that can be made in this process. You'll also notice, though, that oxygen is the acceptor in this particular example. If oxygen is the acceptor of the electrons at the very end, this is called aerobic respiration, meaning that oxygen needs to be present. When the oxygen accepts the hydrogens and the electrons, it gets converted into water. So this is just a picture form that shows you how the electron transport system works, and this system is found in the plasma membrane in prokaryotes. All right, so here's another example where it's showing you the bilipid membrane here. It's showing you that these proteins that are embedded through where the electrons are moving through, which also causes hydrogens to be transported. At the end, we have what we call the ATP synthase. This is an enzyme because it ends in ACE, and it's going to help make the ATP. We also see that there are some important other types of enzymes who are going to help with this. All right, there's what we call the NADH reductase, the succinate dehydrogenase, the cytochrome reductase, and also cytochrome oxidase. Now, cytochrome oxidase is one of the tests that we can look for when we look at the oxidase reaction. Okay, and you'll learn about this in lab, that we can actually take those particular organisms, we can rub it into the filter paper, and we can add the reagent for oxidase. If it changes color and it changes to purple, this tells us that this enzyme, cytochrome oxidase, is present. And if it's present, this means that it is going to be able to do oxidative phosphorylation. If it's not, then it may not be able to do that process. Okay, so this biochemical test is called the oxidase test, and it's going to detect the presence of that cytochrome C, the cytochrome oxidase. So organisms generate ATP via various metabolic pathways. They can use the substrate level phosphorylation only, they could use oxidative phosphorylation only, or they could use a combination of both. So if we look here, this is going to compare three different types of respiration. So the first is aerobic respiration. In aerobic respiration, this means that they need oxygen present, so molecular oxygen has to be there, and this is going to be the final electron acceptor. When this happens, it is a combination of substrate level and oxidative phosphorylation. For eukaryotes, we see that this can create 36 ATP, but for prokaryotes, it can actually create 38 ATP. Another form is anaerobic respiration. Anaerobic respiration requires anaerobic conditions, which means that they don't have oxygen present. So this is going to be where they don't have oxygen, or they don't want oxygen to be there. This is usually going to have an electron acceptor who is inorganic, that's going to accept the electrons that's not oxygen. This is going to also be a combination of substrate level and oxidative phosphorylation. Now, the issue with this, with the ATP, so this is going to be very variable. It's going to be fewer than 38, so it's not going to be as good as aerobic, but it is going to create more than 2. Now, fermentation is the one that is the least productive. Fermentation can have aerobic or anaerobic conditions. The electron acceptor is an organic molecule, and this one's only going to do substrate level phosphorylation. So this is going to create only 2 ATP, which is not a very efficient form of creating ATP. So when we're looking at this whole idea of carbohydrate catabolism, we're going to be breaking down carbohydrates. And more specifically, the cells really prefer glucose. Glucose is the most common carbohydrate used as the source of energy to create ATP. It's the easiest one to break down, therefore the cells want to use it. Cells are very lazy when it comes to creating this stuff, and so it wants to take the easiest path normally. However, other sugars could also be used. It's the oxidation of organic molecules to get energy in the form of ATP. Aerobic cellular respiration is where the final electron acceptor is oxygen. This oxygen is going to then help produce water, and water is going to be one of the products that comes out of the reaction. All right, so water is going to be one of the products. And if you'll notice from the chart previous, we saw that it can create 36 ATP for eukaryotes and 38 ATP for prokaryotes. Now, when we're looking at aerobic respiration, it's going to take all of those steps that we talked about earlier and it's going to put them all together. So we're going to see glycolysis is going to take place. This is not going to require oxygen. This is the breakdown of glucose, and it's going to produce 2 pyruvates. We do see that it does create 2 ATP molecules. This is going to be through substrate level phosphorylation, and it also creates 2 NADHs. These NADHs are going to be reused to generate ATP via oxidative phosphorylation in the electron transport system. We then see step 2 is what we call the preparatory stage. During the preparatory stage, oxygen is required to be present, but it's not going to be used. So oxygen is kind of like the ticket to get into stage 2, but it's not going to be used yet. In this case, though, we're going to convert the 2 pyruvates into 2 acetyl groups. This is going to allow them then to go into the next stage, which will be the Krebs cycle. In this preparatory stage, no ATP is made at this point, but it does create 2 NADHs. And recall that these 2 NADHs are going to be used later on to create ATP through oxidative phosphorylation in the electron transport system. The next step is the Krebs cycle. The Krebs cycle is also known as the TCA, or the citric acid cycle. One thing to note, the Krebs cycle does turn twice per glucose molecule. Since glucose got broken into 2 pyruvates, we're going to see that it's going to turn twice. Oxygen is still required to be present here for the majority of the reaction, but it's still not used. Now, CoA, or coenzyme A, is going to carry the acetyl group to the Krebs cycle. So this is going to be acetyl plus oxaloacetate, which is going to yield citric acid, thus the name that it gets, the citric acid cycle. This cycle is then going to generate 2 ATPs. These ATPs are going to be made through substrate level phosphorylation. We also see it's going to create 6 NADHs and 2 FADH2s. Remember, though, that these NADHs and FADH2s are going to create more ATP once they go to the electron transport system. Now, the ETS is going to be coupled with that chemiosmosis. The chemiosmosis is going to allow us to do the oxidative phosphorylation. This is where the aerobic process really happens. Oxygen is not only required, it is now used. It's going to be used to accept the electrons and the hydrogens, and water is going to be created. There are special proteins that are going to help this process happen. One example is the cytochrome oxidases. These guys are embedded in the plasma membrane of the cell when we're looking at a prokaryotic cell. Input of oxygen atoms is going to happen here. So 1 electron into the ETS is going to bring in 1 proton. This is the chemiosmosis. It's moving those chemicals of the protons. Since 10 ADHs and 2 FADH2s come in, they are going to be responsible for allowing this process to create a lot of ATPs through oxidative phosphorylation. The 10 ADHs are going to help generate about 30 ATPs, and the 2 FADH2s are going to help generate about 4 more. These electrons plus the hydrogens plus the oxygen is going to then create the water. All right, so this particular table is taking this whole idea of glycolysis, the prep stage, and the Krebs cycle and putting it all together. Okay, it's showing you how many are made, and it also is showing you, too, what is going to happen in the electron transport system. And this is going to add up those ATPs, where it gives you 38 potential ATPs for a prokaryotic cell. Now, a note here is eukaryotic cells can also generate a large amount of ATP, but they're going to generate 36 ATPs from 1 glucose molecule, not 38. In anaerobic cellular respiration, the final electron acceptor is some sort of inorganic molecule. This is something other than oxygen. Some, but not all, reactions in the Krebs cycle or the ETS can run without oxygen. So this normally is going to produce less ATPs. Now, it could still produce 36, it could still produce 35, it still could produce a large amount, but it is going to be less than the aerobic respiration due to oxygen not being present. Now, some of these inorganic molecules that are used other than oxygens are things like nitrates, sulfates, and carbonates. Could potentially be those electron acceptors. Now, in a lab, if we're dealing with an anaerobic bacteria, we see that they are going to grow a little slower than we see with the aerobic. Okay, and this is due to the less ATP that they are producing. The last example we saw here was fermentation. Now, fermentation generates energy from various organic molecules such as sugars, but also amino acids and purines. Organic compounds serve as the final electron acceptor, and this is what makes it different than anaerobic respiration. It is an anaerobic process in that no oxygen is required, but oxygen can be present. All right, it doesn't have to be there, but it could be there. This process does include glycolysis. Okay, so the same process with glycolysis is going to happen. It does not use the Krebs cycle or the ETS, the electron transport system. Therefore, since it's only using glycolysis, we're going to see that it's only going to create 2 ATP. Now, with this though, guys, it does create some higher energy end products. These end products could potentially be broken down more, but they don't have the means to do this. These could be things like lactic acid fermentation or alcohol fermentation. So with fermentation, it is only substrate level phosphorylation, and we see that it only is going to create 2 ATP. So the end products of fermentation, if we do a chemical analysis of the end products, this can help us identify the microbes. This can also help us identify pathogens in a clinical specimen, depending on what is present. All right, so this just gives you some example here that if lactic acid is what's actually being produced as an end product, this could be Streptococcus, Lactobacillus, or some sort of bacillus species. However, if we see that the end product is more of a butyric acid or butanol or acetone, this would be maybe a Clostridium species. And so this whole process of the end products lets us be able to evaluate what type of fermentation is taking place and potentially what organisms we are dealing with. So how do we do this? A lot of times we end up doing this through some sort of pH indicator. In this case, they're going to use phenol red. It starts out as being red, but under acidic conditions, let's just say that they've made an acid through this process, it turns phenol red from being red to yellow. The tube on the right has been inoculated with E. coli. Note the positive result of both the acidic production, which means it's yellow, but also if you'll notice in the Durham tube, that little tube on the inside, there's also gas produced in there. There's a bubble there, and this lets you know that it was undergoing fermentation. The tube on the right is an uninoculated tube, so that's what the tube would look like to begin with. It had the red color with no gas bubbles in the Durham tube. Now, fermentation can be used in the industrial setting quite often. If you look at the idea of the fermentation of ethanol, ethanol is used a lot of times to create beer and wine and also fuel. If you've ever, gassed up your car, you'll notice that it has ethanol present. So it depends on what the starting material is. If it's starch or sugar, this is going to do with the beer and wine. Agricultural waste, this would be more of your fuel. All right, so it shows you the microorganisms that potentially could be used. We have lactic acid, could be used to make cheese, yogurt, bread, certain kinds of bread like rye, sauerkraut, summer sausage, and it depends again on the microorganism and what the starting material is. There's lots of different industrial uses for the different types of fermentations. So different catabolic pathways are used by bacteria to oxidize organic substrates. So it's whether they're going to use oxygen, they're going to use, if oxygen is not present, they're going to use organic molecule or an inorganic molecule. It just depends on what that organism is able to do. Enzymes must be present and active in order to drive these chemical reactions, though, in these metabolic pathways. More ATP are generated by bacteria via aerobic cellular respiration than anaerobic cellular respiration. Even less is generated through fermentation. When lower amounts of ATP are generated, this is going to be equated to slower growth in the lab and even slower growth in the environment. Now, sugars are not the only thing that could be used to potentially create energy. We also see that lipids and proteins can also be broken down. Most bacteria are not limited to a single catabolic pathway. They have alternative pathways which allows them to adapt to different environmental changes, especially if sugars are not present. All right, and so when we look here, you can see in this pathway that, of course, glucose is their primary source. However, if fats are present, we could potentially use them to go into the Krebs or citric acid cycle, and the same thing over here with proteins. Now guys, please read the section over the biochemical tests and the bacterial IDs. This information will be presented later in one of your labs. I believe it is lab 9. But one thing to note is that you should read over this because there are different chemical reactions there's different biochemical tests we could do to potentially see what organisms we're dealing with. Now the last little thing we want to look at is what we call metabolic diversity. And we hit a little bit of this when we were talking about the different organisms way back in Unit 1. So when we look here, there's what we would consider the phototrophs versus the chemiotrophs. Phototrophs are going to use light as their energy source, whereas chemiotrophs are going to use chemicals as their energy source. We also see that there's autotrophs versus heterotrophs. Remember that the autotrophs are the self-feeders. They use inorganic molecules to make organic molecules or food. These guys are going to be able to either do photosynthesis or chemiosynthesis. On the other hand, heterotrophs are going to rely on organic molecules for metabolism, so they have to actually consume their food. Photoautotrophs are examples like plants, algae, or cyanobacteria, and chemioheterotrophs, meaning that we take chemicals to eat in there. These are going to be animals, most fungi, protozoa, and bacteria. So guys, if you look here, this is just showing you the nutritional classification of organisms. It shows the energy source, whether it's chemical or light. So you have your chemiotrophs, you have your phototrophs. With the chemiotrophs, we see that there's the carbon source. The carbon source is going to either come from organic compounds or CO2. So you have the chemoheterotrophs, and you also have the chemoautotrophs. When you're looking at the chemoheterotrophs, this is the area we're going to focus mostly in on, because this is going to contain the bacteria, the animals, the fungi, the protozoa that we're looking at here in microbiology. So when you see here, you can see if oxygen is the final acceptor, this is all animals, most fungi, protozoa, and bacteria. But if oxygen is not the final acceptor down here, you can see that the organic product compound is that. We're going to see that it's through fermentation. This would be an example of a streptococcus. On the other hand, if an inorganic compound is the electron acceptor, this would be something like the Clostridium species. We also see then over here the phototrophs. The phototrophs could either be photoheterotrophs or photoautotrophs. And so these give you some examples. So if we're looking at the photoheterotrophs, this is going to be like your green non-sulfur bacteria, purple non-sulfur bacteria. If you're looking at the photoheterotrophs, if they use water to reduce the carbon dioxide, these are going to be oxygenic photosynthesis like plants, algae, and cyanobacteria. If it's anoxygenic photosynthetic bacteria, this would be green and purple bacteria. So guys, here highlighted in the red, these are the ones we're going to spend most of our time looking at, and most of them are under the chemioheterotrophs group. All right, so that covers our chapter 5. If you have any questions or concerns, please let us know.